Okay, welcome everybody to today's Friday noon seminar, and it is a pleasure to introduce Dr. Mark Lubberink from Uppsala University, and he's currently professor of molecular imaging at the university's positron emission tomography center. He did study technical physics at Eindhoven University of Technology in Eindhoven, Netherlands, and received his PhD from Uppsala University in 2001 with a thesis on quantitative PET with long-lived isotopes. After his thesis, he worked as a postdoctoral fellow and medical physicist at the Department of Nuclear Medicine and PET Research at VU University Medical Center in Amsterdam. And since 2010, he has a position as senior medical physicist at Uppsala University Hospital. And he is also head of diagnostic medical physics group since 2015. And his main research is quantitative PET methodology and tracer kinetics, aiming at clinical implementation of quantitative PET methods. He's also a co-founder at Medtrace Pharma, aiming to make quantitative blood flow measurements with oxygen-15 water PET and make this available for clinical use. Also, I always tell my students that you will be doing your PhD or master's with stay with you for life. And this is the case with me and Mark. So we did our PhD with the same supervisor. So you could say that he's in my academic family and a big brother, academic big brother that we are following each other through life. I think you'll never get rid of me. So please, Mark, go ahead.
Thank you, Shirin, and thank you very much for inviting me to give a talk at this lunch seminar, which for me is a dinner seminar. So if I all of a sudden disappear, I've fallen because I'm hungry, I guess. I think my kids would want me to be on the way to buy pizzas right now, but they're gonna have to wait for an hour more. So yeah, Shirin, thanks for the introduction. I'll be talking about clinical implementation of what I would say real quantitative PET methods. I'm sure you're all familiar with the traditional FDG PET scans, which are used a lot. I think 98% of all PET scans done in the world are FDG PET scans to find tumors. But there's so much more to PET that we can use, and that's what I would like to talk about. I don't think I have to show my disclosure because Shirin already talked about that. So I'll start with a little background on Uppsala, and then I'll talk about quantitative imaging in general, and I'll give two examples of applications of real quantitative PET that we have partly developed and are using now in the hospital, not only in Uppsala, but on many other sites as well. Myocardial blood flow and imaging of dopamine transporter availability and blood flow in patients with Parkinson-like diseases. This is a map of the world with Uppsala right in the center, so you all know that's where you should be. And actually, in the 1980s, Uppsala was also one of the centers, at least of the world, when it came to PET. If you wanted to buy a PET scanner back then, you could buy it two different places. You could buy it in Knoxville at CTI PET Systems, and or you could buy it at Scanditronics in Uppsala. So CTI was eventually bought by Siemens, and Siemens was smart and left the people there doing what they were good at. So they are still building their PET scanners in Knoxville. A couple of years later, I think in the early 90s, Scanditronics was bought by GE, and GE Healthcare, of course, thought, okay, we'll do everything in Milwaukee and we'll force the Swedes to move there, and nobody wanted to move. So in the end, essentially, they bought nothing, had to start everything from scratch, but they closed down the PET scanner factory in Uppsala. The PET center in Uppsala is actually started already the work with PET in the early 1970s when Professor Bengt Långström started a carbon-11 chemistry program. So what we are working on is still very much based on the development of new tracers labeled with carbon-11. The first PET scanner Scanditronics, obviously, bought next door, was installed at the hospital in Uppsala in 1982. And in 1991, the PET center that we're still using now was established when the cyclotron was bought. A couple of years later, we got a preclinical PET facility, which now also has PET-MR for preclinical studies. And actually, for those of you familiar with PET, the first PIB scan showing amyloid plaques in the brains of Alzheimer's disease patients was done in Uppsala in 2001. So that's one of the highlights, I guess, of this time. This is used clinically all over the world now. We had a few dark years when the PET center was taken over by GE, but now we're simply an academic site again. And I think we've done more than 25 first-in-man studies here, which is quite remarkable, I guess. We've had more than 100 traces in humans, and on a routine basis, we have about 15 traces in clinical use. And one of the reasons for that is that the Swedish legislation is very forgiving for using non-registered radiopharmaceuticals for diagnostic use, as long as you produce them according to the rules of good drug production. And there is a medical doctor that wants to use them for diagnostics, we can use them in the clinic, which is a very different situation than in the United States. I don't know about Canada, but I think it's more like the US than like Sweden in this respect. So this is an aerial photograph of the hospital in Uppsala. And if we zoom in a little bit, we actually have our nuclear medicine work spread out over two departments. We have down here the PET center, which has a cyclotron, two digital PET CT scanners, and a PET-MR. And that is why the chemistry labs are. So that's where we do all our work, which is non-FDG, which I'm going to be talking about in the rest of my talk. At the top of the drawing here is a new building in which the nuclear medicine department is housed. And that's where we will do all our standard routine nuclear medicine scans, so FDG PET, for example. As I said, the cyclotron was bought in 1991, so it's 30 years old now, and it's time for something new. So we actually bought a cyclotron, a new cyclotron, already 2 years ago, I think. But as I think it's the same in Canada, hospitals in Sweden are publicly funded, and it's one thing to buy a machine, but that somebody also has to build a building to put it in, and we still don't have that, but we're working on that. I think we have a very nice place to do PET from a chemistry and radiation physics perspective. It's a very compact building which was built specifically for this, which is very nice. So we have a clinical floor where we have two PET CT scanners and a PET-MR, and immediately below that we have a chemistry department where we have a cyclotron and we have our GMP tracer production. As I said, we bought a new cyclotron that's going to be placed just outside the building, so we're actually going to expand our chemistry facilities by about a factor 2. So we'll get the cyclotron outside the building, we get new chemistry labs, and it's about time for an update after 30 years. I think the Uppsala PET work is a bit special, as I said, because we are using so many different traces in the clinic, not just FDG. This is everything we use, which is not FDG. And you see there's a wide range of different traces that we're using there. And I think what is special here, and what I'm going to talk about more, is that in some of these we actually try to utilize the real quantitative capabilities of PET. And so not just making images, but actually trying to calculate biological parameters behind the PET scans, which give us more information. And we have then about 25-30 different traces for research that we can use at any time right now. Of course, this graph is from 2017. The last 2 years, not a lot of research has happened because of COVID, but we're starting things back up now. So I hope we will not have a fourth or fifth, or I lost count, wave of COVID that will stop this again. The aim of our work in PET, and that is then from a clinical point of view, from a physics point of view, and from a chemistry point of view, is we want to develop tracers and methods for improved diagnostics and treatment selection. So the use of PET essentially as a part of precision medicine, finding the right treatment for the right patient. One of the things that we also do a lot of work on is, and that's not PET, but more traditional nuclear medicine, obviously, patient-specific dosimetry-guided radionuclide therapy of neuroendocrine tumors and prostate cancer. And a third aim is what I'm talking about here, methodology development for quantitative functional imaging with PET, PET-MR, PET-CT, and automation so that we can use it clinically. And I mentioned the word quantitative already a number of times now. And so I'm sure you've all heard this. PET is a quantitative imaging method that measures biology. That sounds very nice. But my question then is, so why doesn't anybody do that? All we do in the clinic is just make images, and we don't actually measure anything. And we could do that. For those of you not so familiar with FDG, F18 labeled deoxyglucose, this is a schematic drawing of what happens to FDG when it goes from plasma into the cells and enters the first step of the glucose metabolism. So on the left-hand side, we have FDG in plasma, and then it is transported into the cells by the GLUT1 transporter. After that, we have intracellular FDG, but it's still just FDG, the intact PET tracer. But then the enzyme hexokinase converts it to glucose or to FDG6-phosphate. And FDG6-phosphate was essentially then, that essentially was a lucky shot when they started using FDG. It turns out that when FDG is converted into FDG6-phosphate, the cell doesn't know what to do with it anymore. So it stays where it is, and it doesn't enter the rest of the glucose metabolism. And that gives us these nice FDG images. We get a very high uptake because any FDG that enters the cell and that is converted to FDG6-phosphate actually essentially stays there. So we get an accumulation of the radioactive fluorine, and we get high contrast images that can show us where tumors are because they use more sugar than other tissues in the body, except for the brain, I guess. And with us being physicists, we can translate this schematic drawing here into a mathematical model that describes what actually happens to FDG when it enters tissues. So this is a compartment model. This is called an irreversible two-tissue compartment model, which mathematically describes with two differential equations the transport of FDG from the plasma to the tissue, where it is still free FDG, and then the first step of the metabolism. So we have three rate constants. K1 describes the transport of FDG from plasma to tissue. K2 describes clearance from tissue of intact FDG. And K3 describes the first step in the metabolism of glucose. And we can solve these differential equations, and then we can write down an equation which describes what we could measure over time if we injected FDG in the scanner and then scanned for an hour. Then this equation would describe the activity concentration that we would measure in a tumor over time during the course of the PET scan as a function of these rate constants, K1, K2, K3, a blood volume parameter, VB here, which we need because we can never draw a region that doesn't include blood vessels, and the spatial resolution of PET will always mean that each pixel will also include blood vessels, and as a function of the plasma concentration over time. So we need to have that to solve this equation. And based on this, we can calculate what we call the net uptake rate of FDG, the Ki value. And once we have that, we can calculate glucose metabolism. We multiply this net uptake rate of FDG in milliliters per cubic centimeter tissue per minute with the glucose concentration in plasma in micromoles per milliliter. And then we divide it by something which is called the Lund constant, which is a factor that accounts for the differences between FDG and actual glucose, which is a fixed number. So if we would do this, we do a dynamic PET scan, measure over time, we can actually calculate glucose consumption in each gram of tissue. So the amount of glucose in micromoles that is consumed in each gram of tissue per minute. So this is physics, right? And it's biology. We can actually measure biology. And if we do that, this is a dynamic PET series over the shoulder area. And you see in this white circle here, you see the slow accumulation of FDG. This is actually not a tumor. This is brown fat that is activated, but the principle is the same. So this is a dynamic scan. We draw a region, which gives us the black dots here, which is the PET data. We have the plot curve, which we either base on a region of interest in our PET image over a large blood vessel, or we take blood samples. And then we can fit our equation that you see at the bottom of the screen here to the measured PET data. And when we have a good fit, we basically have found the K1, K2, and K3 values that describe our measured PET data best. We can also do that at the voxel level. And if we do that at a voxel level, we get the image at the bottom here. But if we would just do a static scan after an hour, we get the image at the top. So a qualitative or semi-quantitative image is what we get clinically. What you see at the top here, the real quantitative image giving glucose metabolism is at the bottom, and they're not that different, right? And at the top, you see the standardized uptake value, which is the activity concentration in each voxel normalized to the injected activity per body weight. And that's a simplified measure of quantification. And it has been shown already 20 years ago that measure actually correlates very well to the true glucose metabolism. So for FDG, actually just doing these standard scans that you see here on the right-hand side, 1 hour after injection, and then calculating these HOV values, gives us numbers that correlate very, very well to the true biology in most cases. So why don't we use the real quantification measuring glucose metabolism? Well, there's a number of reasons for that. For FDG, it's not really needed most of the time. Other than that, it takes too much time because our PET scanners only cover 20-25 centimeters in one bed position. So if we want to scan dynamically, we can only cover 25 centimeters at a time. If we would want to scan the whole body, we have to bring the patient back every day for a week, and each day scan a different part of the body. So we can't really do that, right? That would be clinically not very useful. We need the blood data, and preferably for accurate quantification, we want arterial blood data because it's tracer in arterial blood that can enter the tissue. And you need one of us, right, to analyze this data, because very often there is no software available that you can just buy off the shelf that does this. So you need somebody that can analyze this dynamic PET data. And that's the reasons why this is not used a lot. But sometimes we miss out on a lot of useful information in this scan, or we can get more information from one PET scan if we actually quantify things than we otherwise get from a number of different investigations. So actually using these quantitative methods could improve patient comfort because we don't have to get the patient to the hospital as many times. So there are actually a number of applications where we would really like to actually use quantitative PET. But it's hard, right? If you do a real quantitative PET study, like we do in research, with a very selective tracer, you're probably going to need cyclotron to make your radioactive isotopes. You need a good PET scanner, obviously. You need to do a dynamic scan, which takes a long time, usually 60 to 90 minutes to get enough data to be able to fit your model. We need to take blood samples. We actually have to analyze these blood samples for how much of it is the intact tracer and how much is radioactive metabolites that still carry the radioactive label, but that are not going to enter the tissue. That's probably the most challenging part, I think, of a dynamic PET scan. You need to apply a tracer kinetic model, like the one I showed you for FDG. This is a more complicated one. Do some fitting, and then in the end you get some numbers, and you can either get those for a region or you can get them at the voxel level. But this is a lot of work. This is complicated. So if we want to be able to use this clinically, we need to make things simple. We need to make sure that us physicists are not needed to do this, right? We need to make ourselves out of a job, basically, but then we'll find something else to do, obviously. So one application where it's actually very useful to perform real quantification is in the measurement of myocardial blood flow, and we can do that with that. Traditionally, if a patient comes to his physician with chest pain, one of the first investigations that is done is a SPECT scan. And in myocardial perfusion SPECT scan, technetium sestamide is injected first at rest, and then we either let the patient bike in the scanner, or we give a pharmacological stress agent that stresses the heart, and we do a second scan. And what we're looking for is then defects in the stress scan which are not visible in the rest scan. And then we say that this patient has reversible ischemia, which is usually caused by stenosis in the coronary arteries. If this defect is visible in both the stress and the rest scan, then probably the patient has had an infarction. But most patients that come to the clinic, they have chest pain because their heart just can't do enough work when they are exercising. And these SPECT images are qualitative. So they don't actually give us any numbers about how much blood that perfuses the myocardial wall, the heart wall, per gram per minute. They just give an image, and usually that is shown as this is called polar plot. So the heart is shaped like a cone, right? And essentially you just flatten it, so you make the heart look like a dartboard. So in the middle you have the apex of the heart, and at the edges you have the basis of the heart, where the blood flows out into the aorta. Now this patient here looks completely normal in a SPECT scan, but she is not healthy. But you couldn't see that on the SPECT scan. And this patient later on got a PET. And this is a 65-year-old female with chest pain, well, the SPECT scan looked normal, so then these doctors want to move on and test something else. So this patient got an 015 water PET scan at rest and after pharmacological stress. And if you look at these images, and now it's not one of these dartboard images or polar map images. Now it's a short, on the top row you see short axis images of the heart. So if you have this cone again, you cut through it so that your heart looks like a row of nice donuts. And these images also look completely normal. It's a nice homogeneous circle without any defects in it. The top row here. Let me get the let me get the pointer. The top row here are images at stress. So when we try to maximize the work, we try to make the heart work as hard as possible. The bottom row images here is at rest. So when the patient is just resting. The other images are the other orientations through the myocardial wall. They look normal. But we know, based on earlier studies, that during rest, a normal blood flow of through the heart wall is about 0.5, 0.6 milliliters per gram per minute. And if you look at the color scale here, corresponding to the rest scan, yeah, well, that's about what we see here, right? But at rest, in any healthy person, the heart should be capable of increasing its blood flow by at least a factor 3 or 4. So what we expect is that during stress, the blood flow should at least be 2.3 milliliters per gram per minute. And if you look at the stress color scale here, you can see that it doesn't reach that at all. It's only about 1.5. So this is a quantitative image. This is not a raw PET image. These are images where the pixel values are not becquerels per milliliter or millicuries per milliliter, but the color scale here is blood flow in milliliters per gram per minute. So we perform pharmacokinetic modeling at voxel level to get quantitative values. And this shows actually that this patient is very sick. There are stenosis in all 3 blood vessels, all 3 major coronary vessels, which means that the entire heart is poorly perfused. And you cannot see that in a qualitative image. you have to do a measurement and quantify it. And we can show it in the same way as these polar plot images that I showed for SPECT before, but now with a color schedule that is no longer qualitative, but it's actually quantitative. We do this with O15 water in Uppsala and a number of other places. And O15 water is considered the ideal tracer for measurement of blood flow. And that is because water is water, it's freely diffusible. All blood vessel walls are completely permeable for water. It's also metabolically inert. Any water that you inject will always remain water. Nothing happens to it in the body. So the only thing it does, it flows with the bloodstream. And an advantage of water is that it is a very high extraction. So the PET signal, the blue line here, is linearly proportional to the myocardial blood flow. And that's not only in the heart, but also in other tissues. The PET signal is proportional to the flow. So we actually measure flow directly. I say the PET signal, what I mean is the clearance rate of the tracer, the uptake rate of the tracer from the plasma to the tissue. There's a number of different tracers that we can use. This SPECT agent, systemibly that I started talking about, has a curve that is the purple line that you see behind the green dotted line, green dashed line here. But it has a much lower extraction, which makes it less accurate. And I'll show you an example of that later on. There are also other alternatives for PET. You can use ammonia labeled with nitrogen 13, which is also a very good flow tracer, almost linear signal. What is most used with PET actually is rubidium 82, which is basically just as bad as sesame SPECT when it comes to the relation between the PET signal and perfusion. But it's very easy to make because you can buy a generator and you don't need a cyclotron to produce it. It's very expensive though. So everything sounds perfect, right, with water, perfect for quantification, but it has a short half-life, 2 minutes. So you need a cyclotron to produce it, and it needs to be nearby. So you have to have that within, let's say, 100 meters from the PET scanners. Otherwise, the distance becomes too far. You have to make the tracer run to the PET scanner. It becomes too hard. And because water is freely diffusible, what goes into the tissue also leaves the tissue at the same rate. So you cannot get any just uptake PET images with O15 water. It will distribute homogeneously through the body after a while. So we need to quantify it, but that is difficult, or has traditionally been seen as difficult. So I'll show you an example of this no uptake image thing. If we have here 3 different PET traces, on the left rubidium, and at the center ammonia, and at the right O15 water. And if you pay attention, and unfortunately the images disappear at the end, but I'll play it one more time. You see that for rubidium and ammonia, you start to see a donut after a while, right? So you get a qualitative image of the distribution of the traces to the heart. But for O15 water, if you look at the right-hand side, after about 4 minutes, the tracer is almost homogeneously distributed. So you don't get an image that will tell you anything. You have to do calculations to get any use of this. How do you do that? So we do a dynamic scan, about 4 minutes. And what you can see here is that we inject intravenously. After about 15 seconds, all of the radioactivity is in the right ventricle of the heart. 10 seconds later, it has passed through the lungs and is in the left ventricle of the heart. And then after about 2 minutes, it's basically everywhere. There's the activity concentrations in the heart wall and in the blood are the same. Traditionally, we draw a region of interest over the heart muscle. A bit hard to see here, of course, because the radioactivity is everywhere. So we get a time activity curve showing the activity concentration in the heart muscle over time during the scan. And you have to draw regions in the left ventricle and the right ventricle to get the blood radioactivity concentrations. So we have those here. Then you put those into a model. And just as I showed you before, you fit it until you get until you get a fit that describes the measured PET data well. But then, of course, you only have one number for the whole heart. And what we want is actually get these images that I showed you before. We want to have quantitative images that show us perfusion at the voxel level. We can do that. We can implement these models in a bit more clever way, and so we can do them really fast at the voxel level to make parametric myocardial perfusion images. And if we did that in the well, let's say early 2000s, then we got an image like this. And of course, this looks like crap. No medical doctor would ever want to base any diagnosis on images like this. And the reason for that is that the PET scanners 20 years ago had such a high sensitivity, poor count rate, lower sensitivity, sorry, and poor count rate performance that we had very noisy data. And if you want to fit very noisy data with a model like this, you'll get very uncertain parameters. So these images were very noisy. And even like in this case, when they were filtered with a like 15mm filter, the image was not useful for any clinical diagnosis. And that's what gave radioactive water a bad name, because everybody says, well, you can't get any images. It has a good name because it is the standard for measuring blood flow, but medical doctors want the images, right? The radiologists want images, nuclear medicine physicians want images, and we didn't have those. But if you do this with some clever mathematics and implementation of these models on a modern PET scanner, you get an image like this, like the ones I showed you before. So actually nowadays, we can do this modeling at the voxel level, so we can quantify myocardial blood flow, and we get images that show us myocardial blood flow at the voxel level. So we can get images that give a visual interpretation, and we also get the numbers that give us the actual blood flow in milliliters per gram per minute. Of course, we can also do that for these other tracers. And these images will show up here in a second at the bottom. So we can make quantitative myocardial blood flow images like this also for these other PET tracers. For some reason, it's a bit easier for O15 water because it has a very simple model. But I won't get into that now. But this works. So once we do the quantification at the voxel level, these tracers essentially become equivalent in terms of how you can read them. You get an image that shows you the distribution of the blood flow, you get numbers, and the advantages with O15 water is that these numbers are very accurate. This is an example of a different patient. Again, the patient got a SPECT scan. Patient had chest pain, got a SPECT scan, SPECT looked completely normal. Now patient got a PET scan and with O15 water. At the bottom, you see the rest polar map. At the top, you see the stress polar map. And as you can see, although the SPECT looked normal, the PET did not. And this patient went to the cath lab afterwards and got an invasive angiography. And indeed, there was a stenosis in one of the coronary arteries, which corresponds to this area with reduced stress perfusion in the PET scan. So how come you can see this in a PET scan and not in the SPECT scan? Well, the reason for that are these curves that I showed you before. The response of O15 water to blood flow is linear, where it is not for systemic SPECT. And in this patient, the reduction in perfusion in the inferior part of the heart here is not very large. It's probably somewhere around where this red arrow is here, and where the red arrow is at a flow of about 2, which is a critical value because it's close to the limit of healthy, not healthy. Any change in blood flow will correspond to a similar change in the signal that we measure with O15 water. But for a technetium system, it doesn't. This curve is not proportional at all. It actually flattens, which we call a roll-off effect. So at these flow levels, the SPECT tracer is just not sensitive enough to pick up small variations in myocardial blood flow. So this is also a reason why PET is more accurate in the diagnosis of myocardial ischemia. And this is not only a problem for SPECT, but also for PET traces, for the other PET traces that have a low extraction, like rubidium here. So there are, since there's mostly physicists in the audience, I'll show some more equations here. I showed you before the solution of the FDG model. This is a solution of a typical simple single tissue compartment model that we use for most PET traces. So we have a parameter K1 again that describes transport of the tracer from plasma to the tissue. And we have a parameter K2 that describes clearance from the tissue. And this K1 for traces like ammonia and rubidium is what we use as a surrogate for the blood flow. Now, water is a little bit different because for water, what goes in must go out. We don't have any accumulation of water. So actually, we see the flow both in the uptake rate and the clearance rate. And what we normally choose to do is that we say, okay, we know this number here, the distribution volume of VT, that is the water concentration in tissue. We know that very well for myocardial wall, that's about 0.9 grams per cubic centimeters. So we actually fix that. And then we can choose whether we want to determine the flow from the uptake or the clearance rate. And they should be the same, right? If what goes in goes out. But in reality, it's not the same because we have all kinds of physical effects in the PET scanner. We have a limited resolution. And a limited resolution means that we'll get a underestimation of the K1, which is based on the amplitude of the time activity curve that we measure. But it will not affect the K2 because that's the shape of the time activity curve. That's not so much affected by the resolution. And there are a number of other effects that end up in this alpha factor here, which make it very favorable actually to measure the flow from the clearance rate, which you can only do for water. And just to give you a little overview of that, so K1 gives you the average flow in a volume of interest that we draw over the heart, and we call that the transmural flow. The K2, the clearance rate value that we calculate with water, gives you the flow only in the perfusible part of the volume of interest, so in viable tissue only. So that's a little bit different interpretation of these images. So all these other PET tracers measure the transmural flow, the K1, the uptake rate of the tracer. But with water, we can choose, we can either measure the clearance rate or the uptake rate. The discrepancy between them is what we call the perfusible tissue fraction, which includes a lot of different things: motion, partial volume effects, all these things that basically degrade our PET images end up in there. But they can also be a measure of viability. So if you have a patient with an infarction, this perfusible tissue fraction will be very low. So we can use O15 water both for measuring flow and viability in these patients. There's a number of challenges when we do these kind of scans with the PET-CT scanner. And one of those is PET-CT misalignment, obviously. We do a CT scan first, and then we do PET. And for most PET traces, we have a problem when there is a misalignment between the two, because the attenuation correction will be wrong. Another challenge is motion during the PET scan. We'd also like to see, for example, how the use of different image reconstruction methods, so the addition of more or less smoothing, affects the quantitative values that we get. And there's a number of other things that we have to consider. But if we look at misalignment between PET and CT, there's been a lot of publications about that for cardiac PET, misalignment is a huge problem. But those publications are almost always based on rubidium or ammonia data. And we try to simulate a lot of different motions, patients moving up and down in the scanner up to 2 centimeters between the CT and the PET, moving left and right. Then we estimated the effect of those. So we added actual misalignments to clinical data in 10 patients. And then we evaluated what the effect of those misalignments was on both the K1 value, so the uptake-based flow, and the K2 value, the clearance rate-based flow. And the results are actually very interesting because, as you can see here, effects on uptake-based flow, K1, are quite big. If we have a motion, a misalignment where the patient is taking a deep breath before we do the PET scan, we can see that we can have an underestimation of, on average, more than 10% in our K1 values. But it doesn't affect the clearance rate at all. So that's very nice. And the reason for that is, as I said before, any misalignment causes an error in attenuation correction. And that affects the amplitude of the time activity curve in each voxel or in each volume of interest. But it doesn't affect the shape. It's just a scaling factor. And it's the shape that determines the clearance rate and the amplitude that determines the uptake rate. So a huge advantage of measuring myocardial blood flow from the clearance rate is that you are much less sensitive to misalignment between PET and CT. For reference, we added intra and inter-observer variability of blood flow values on the right-hand side here, and you see that the effect of misalignment on clearance-based flow is not much more than the inter and inter-observer variability, whereas for the uptake-based flow, it's much more. So what happens then if the patient moves during the PET scan? That's of course much more difficult. It's very difficult to correct for that. And these patients get this pharmacological stress agent, which sometimes gives them discomfort. So it's rather common that patients move. And we also looked at that. So we introduced motion to clinical scans that we had checked beforehand that the patient actually were lying still very nicely. And what we found here is that, of course, motion is a problem, but as long as that motion is less than about half of a myocardial wall thickness, so about 5 millimeters or less, the effects of motion on the clearance-based flow are also not much more than the inter or intra-observer variability. And this is also a smaller effect than it would be for uptake-based flow values. And then finally, I guess you can almost guess is the effect of image reconstruction method. As I said, if we have misalignment between PET and CT, we have very little effect on the K2, on the clearance rate, because the shape of the curve is not affected, but we have a lot of effect on the K1, the amplitude of the curve. Now, different image reconstruction methods primarily affect the amplitude, right? If we use a sharper filter, a image reconstruction which results in a better spatial resolution, we get higher K1 values because the amplitude of our time activity curves will be higher. But also there, they won't affect the shape. So on the right-hand side here, we plotted correlation coefficients between myocardial blood flow values for different parts of the heart between different image reconstruction methods, ranging from an iterative reconstruction with only one iteration, so a very bad resolution, bad contrast reconstruction, to a penalized reconstruction, which gives a resolution of about 3 millimeter in our PET images compared to a image with an 8 millimeter filter, which has like a 10 millimeter resolution. And if you look at the arc square values here on the right-hand side, there is some variation here, but they're all above 0.99. So image reconstruction also affects these values very little. And that's nice, because if we want to be able to use quantitative imaging across different sites, we have to make sure that we can actually compare what we measure in Uppsala to what we measure in Montreal. And then it's very good to have a robust method that is not dependent on image reconstruction, that is probably also not very much dependent on which PET scanner we use. So that is also an advantage of the clearance-based flow. So using these quantitative methods, we can actually do what we would say multi-parameter myocardial blood flow imaging with our 15-part PET. Not just images, but actual measurements of quantitative values. And not only blood flow, but also then viability. We can also measure extraction fraction. So how much does the heart pump at every beat? And we can measure the volume, the mass, the wall thickness of the myocardial wall. And because we have the lungs in the field of view, we can also measure lung perfusion, which can give additional information to the clinician. And we have part of the liver in our field of view, so we can measure liver perfusion, all from a single scan. So this gives so much more information than just looking at an image. I have one other example, which I will go through a bit quicker, because otherwise your lunchtime will be over and my pizza time has come. The brain. I mean, of course, these quantitative methods are not only interested in the heart, they're interested everywhere, interesting everywhere in the body. And an example that I'd like to give there is in the differential diagnosis of patients with Parkinsonism. So these are patients that come to their physician with the typical symptoms of Parkinson's disease, right? Tremors, problems walking. And when the the traditional way of diagnosing this actually is that you start treating the patient with levodopa. If that works, it's Parkinson. If it doesn't, it's something else. That's not very nice for the patients because there's a lot of side effects. So a better option is to do a SPECT scan with a tracer called DOT scan, which binds to dopamine transporters. And if a patient has Parkinson's disease, then the availability of these dopamine transporters in the striatum, these two structures here in the center of the brain, is affected, and we can use this image to diagnose that the patient has Parkinson's disease. But there are like 7, 8, 9 other diseases that give the same clinical symptoms. And some of them also cause a decrease in dopamine transporter availability, but others don't. This scan will look completely normal. So that's why we also want to do an FDG scan, because an FDG scan can tell us more about which of these other diseases the patient might have. Each of these different Parkinsonisms will give a different pattern of hypometabolism on an FDG scan. So we do these 2 scans. But a couple of years ago, we thought, okay, we have a very nice PET ligand, carbon 11 labeled PE2I, which is a PET equivalent of this DOT scan tracer. But it is more selective, so it really binds only to dopamine transporters, whereas DOT scan also has some binding to other transporters in the brain. And as you can see in these images, of course, because we can use PET, the spatial resolution is much better. And we've been using this in many clinical studies, many research studies, hundreds of subjects. And what we usually do there is we do an 80-minute dynamic scan, because now we want to quantify dopamine transporter availability. And then we use a model that describes the kinetics of this tracer in the brain. This becomes a bit more complicated. We have tracer in plasma, which enters the brain. It's still the intact tracer in the brain, and then it can bind either specifically to the dopamine transporter, but usually there's also non-specific binding to other binding sites. This model becomes a bit more complicated, but very often we can actually lump these non-specific and free tracer together, which makes things easier. We also want to avoid using blood data. And in the brain, fortunately, there's very often a region which is similar to our target region in every way, except that it lacks the specific binding. In case of dopamine transporters, we can use the cerebellum for that. And we can use that instead of blood in our quantification. We call this a reference tissue method. So we can set up a compartment model where we actually do no longer have to include the radioactivity concentration in the blood. We can instead use a reference region in the brain, and we can set up an equation that describes our PET data over time, which no longer includes plasma. So that's nice. We don't want to have to take blood samples. And we figured, okay, this tracer is much more specific. And if we do this tracer kinetic modeling of a dynamic scan, we get a number of parameters. One of them is called the binding potential. And the binding potential is a measure that is directly proportional to the concentration of available dopamine transporters. So it's really a number. And this number we can compare between subjects, and we can use it to evaluate treatment or disease progression. So it's a quantitative value that describes the dopamine transporter availability. We also get a parameter which is called R1, which is the relative blood flow. So that is the ratio of the blood flow in each part of the brain to our reference tissue, cerebellum. And we can solve this, at the voxel level. And then again, here you see this DOT scan and FDG image on the left. But now on the right, you see a binding potential image based on this dynamic carbon 11 P2I scan at the top. And at the bottom, you see this relative blood flow image relative to cerebellum. And as you can see at the top, these images give the same information, but with a much better resolution and the added value of quantification for the PET tracer. And at the bottom, we see that this relative blood flow image looks very much like the FDG image, right? So you could actually use the 2 parameters that you get from the dynamic P2I PET scan as a surrogate for the 2 different scans that we traditionally give these patients. And that is good for the patient because it means 1 instead of 2 visits to the hospital. You can't do the DUT scan and the FTG on the same day. Often you can at least not do the FTG before the DUT scan because it will disturb the SPECT measurement. And if you want to do them on the same way, on the same day, it causes more planning, obviously. The radiation dose is much lower. The DUT scan plus FTG is somewhere between 10 and 15 millisievert, whereas the radiation dose of this carbon-11 scan is 2 millisieverts only. And we get more accurate, high resolution quantitative results. Now this is an 80-minute scan. That's of course too long to use in the clinic. So we went on and said, okay, this is a too long scan duration. For DUT scan, a DUT scan SPECT takes about 30 to 40 minutes. So we figured if we can condense this dynamic protocol down to 40 minutes, then this is a real alternative for doing SPECT plus FTG. The way we analyze our research scans requires a co-registered MRI for region definition, lots of manual processing. So the questions that we wanted to answer is, can we do this shorter and can we automate this? We looked at the scan durations, and it turns out that if we at the bottom here you see this on the right, the right 3 graphs in the bottom row show you these relative blood flow images of values, and it turns out that if we go down to 20 minutes of scanning, we still get a very good correlation with the values that are based on the full 80 minute scan. So to get these blood flow values, we could actually shorten the scan a lot. And also, instead of this binding potential, we can use a SUV ratio at 30 to 40 minutes, and that also correlates very well to the binding potential. So we can actually simplify this a lot. And this is the protocol that we're using now. We're doing a 40-minute scan in these patients, and that gives us both a dopamine transporter availability image and a relative blood flow image. And until 3-4 years ago, when we did this DUT scan FTG combination, we scanned several tens of patients per year, maybe, for this question. But since we started doing the PET scans, the neurologists have become really enthusiastic. So now we do hundreds of them, and they've really made this to their own thing. They think it's fantastic that they have the quantification and that they get everything from a single scan. As I said, we try to automate this, obviously. So we want to develop an automatic workflow that takes this dynamic PET data and that automatically processes this and gives us numbers comparing to a normal control database. So one of my colleagues, Charles, who you know, I think, Shirin, yes, he wrote a software that is implemented within Hermes, but we can run it from anywhere, which first does motion correction, because these patients, of course, have a tendency to move. Does a spatial normalization to a MNI space, and then models the dynamic scan using the cerebellum as a reference region. So we get these relative cerebral blood flow images and the dopamine transporter, the availability images. And when it is applied a template, standard region of interest template, it will then give you a set of Z-score values that show the distance in standard deviations between the individual patients that we're looking at and our normal database. And you can also show that in the formal service prediction. So this is very much like Cortex ID and these other softwares that you can use to assess patients with dementia symptoms, but for a multi-tracer approach, multi-parameter approach with a single PET tracer. And we are going to continue this work. We are doing setting up a multi-center study with 3 other universities in Sweden, where we're going to use an F18 labeled analog of this tracer, because of course F18 makes things much easier. You don't have to produce it locally. You can transport the tracer, and then we're going to develop AI methods to automatically do the differential diagnosis based on these images. I have 2 slides left, Shirin, if that's okay.
Yes, please go ahead. I just have to open the door for an engineer that is going to look at my house at something. So continue, I'll be back.
Yeah, good. One thing that, a point of caution that we realized when we tried to do these scans in the PET-MR, instead of on a PET-CT. We scanned a number of patients both on a PET-CT scanner and a PET-MR to compare the quantitative values that got on both scanners. And this is one of these relative blood flow images based on a dynamic PE2I scan that we acquired on PET-MR. And when you compare it to the same image on actually this is a dedicated PET-only scanner, you see that there's actually some remarkable differences, and those correspond directly to the auditory cortex, and we assume that this is caused by the loud noise in the PET-MR scanner. So we realized that it's not as easy as we hoped it would be to be able to combine PET-MR and PET-CT data of these kinds of traces in the same studies. So to summarize, efficient implementation and automation of these quantitative methods allows for clinical use of functional PET, so real quantitative PET in routine clinic. And these methods can be applied in many different applications than what I showed here. I mean, these dynamic brain scans, these methods can be done with any, almost any brain PET tracer. And especially if you want to do this in a PET-MR anyway, you can combine it with a comprehensive MR protocol for neurodegenerative diseases, which also takes about half an hour to do. So it's a good way to utilize the full time in a PET-MR scanner if you want to use it. And then, of course, this can also be relevant in whole body imaging. I know Siemens is really trying to promote this whole body parametric imaging with FDG. And then, of course, when we will get our total body PET scanners, which I guess everybody would be interested in having, we can get this dynamic data for the whole body at once. And it will give us all kinds of new possibilities to look at interaction between the brain and the rest of the body, and to actually quantify the underlying biological processes at the total body level, instead of one PET position at a time. This is the medical physics, nuclear medicine and PET researchers, one of our meetings during the last year. And thank you for your attention. Trying to make shit in a little